Hi everyone, I had page 4 of the lab experiment, so that includes the nitrate broth and the citrate slant. And so I'm gonna bump them into 2 videos because otherwise the internet gets mad at me for the video being too long. So the first test we'll be doing is the nitrate reductase test. And so I have everything up on my computer, but I also have it printed here just to try to make things easier. So as far as aseptic technique goes, we want to have our hair up and tied, goggles, gloves, and apron if necessary. Make sure you wash your hands when you enter lab, wipe down your lab station, all that stuff. And then for this lab, we're going to be using an inoculating loop. And so in the real lab, we'd have 5 broth samples that we're doing, but in this case, we're just going to show you one. So as far as aseptic technique goes, with your inoculating loop and your micro incinerator that's been turned on for 10 minutes, so it's nice and hot, you're going to place your loop in the micro incinerator for about a minute, and then remove it and let it cool for about 10 seconds. If you don't wait that 10 seconds, it'll be way too hot, it can kill off any bacteria. And then we're going to open our source culture, remembering to use that pinky finger so that your loop stays sterile. Once you open that culture tube, you want to hold the lip of it against, or close to, not against, the micro incinerator opening to kill off any bacteria. You only need to hold it there for 2 to 3 seconds. Then you can put your inoculating loop into the source culture, get a good sample. You'll have swished it around, of course, before you opened it so that everything's everywhere. It's not just separated out. Remove your inoculating loop that has your beautiful sample on it. Hold that lid up against the micro incinerator again, 2 to 3 seconds, then replace the lid and you can put that source culture back. Next, you're going to take your samples. In this case, we're just doing the one. Remember, we have 5 in an in-person lab. Again, open it with that pinky finger, hold it in front of the micro incinerator 2 to 3 seconds, kill off any bacteria, and then you can inoculate your sample with this particular source culture. Hold that in front of the micro incinerator, lid back on, hold this inside the micro incinerator, take it out, and you have inoculated your broth samples. And so you want to have everything labeled, of course, especially in this instance where you're gonna have 5 instead of just the 1. And then you're gonna incubate at 37 degrees Celsius for 12 to 48 hours. So then the next time you come into lab is when we're actually going to be able to see if the nitrate was actually reduced. So a couple things we're going to need from our reagent cabinet. We need sulfonilic acid, or nitrate A, and alpha-naphthylamine, or nitrate B. We're going to use 20 drops of each of these into each of our tubes. And remember, every time you open a tube, you should be holding it in front of the micro incinerator to kill off any bacteria maybe hanging out in that lid or on your hands, just to keep things as pure as possible. After you have added your drops, you're going to mix each tube really well. Make sure that everything has kind of had time to travel through the substance. And at this point, if your broth sample has turned red, we do have a positive reductase result. But we're not going to stop there. There is another step to this experiment to make sure we are actually evaluating if it's a positive or negative reductase result. If the broth is not changing color, then we want to add zinc dust, which is our other thing that we're going to grab from our reagent cabinet. So in the nitrate broth cultures that do not have any color change thus far, I'm going to take a toothpick, pick up a few particles of the zinc dust and drop it into those broth samples, and you're going to mix that well. At this point, if the broth turns red, then it's a negative reductase result. If the broth does not turn red, then it's a positive reductase result. From there, you're going to do your standard cleanup, so dispose of chemicals in the properly labeled places, wipe down your entire lab station, put the reagents back into the cabinet where they're stored, turn off your micro incinerator, and you're good to go.